Hello, welcome to Biology, the scientific study of life. Evolution of genes with novel functions. In the evolution of a global gene family, as gene duplication and subsequent divergence produced family members whose protein products performed related functions. Alternatively, one copy of a duplicated gene can undergo alterations that lead to a completely new function for the protein products. The genes for lysozyme and alpha lactalbum are good examples. Lysozyme is an enzyme that helps prevent infection by hydrolyzing the cell walls of bacteria. The lactobumin is a non-enzymatic protein that plays a role in milk production in mammals. The two proteins are quite similar in their amino acid sequences and the three-dimensional structures. Both genes are found in mammals, whereas only the lysozyme gene is present in birds. These findings suggest that at some time after the lineage leading to mammals and birds had separated, the lysozyme gene underwent a duplication event in the mammalian lineage, but not in avian lineage. Subsequently, one copy of the duplicated lysozyme gene evolved into a gene encoding lactalbumin, a protein with a complete encoding alpha-lactobumin, a protein with a completely different function. Rearrangements of parts of gene, exon duplication and exon shuffling. Rearrangement of existing DNA sequences has also contributed to genome evolution. The presence of introns in most eukaryotic genes may have promoted the evolution of new and potentially useful proteins by facilitating the duplication or repositioning of exons in the genome. Recall from chapter 17 that an exon often codes for a domain, distinct structure or functional region of a protein. We have already seen the unequal crossing over during meiosis can lead to duplication of a gene on one chromosome and its loss from the homologous chromosome. Notice this figure. By a similar process, a particular exon within a gene could be duplicated on one chromosome and deleted from the homologous chromosome. The gene with the duplicated exon would code for a protein containing a second copy of the encoded domain. This change in the protein structure could augment its function by increasing its stability, enhancing its ability to bind a particular ligand. Or altering some other property. Quite a few protein coding genes have multiple copies of related exons, which presumably arose by duplication and then diverged. The gene encoding the extra structure or the extracellular matrix protein collagen is a good example. Collagen is a structural protein with a highly repetitive amino acid sequence which is reflected in the repetitive pattern of exons in the collagen gene. Alternatively, we can imagine the occasional mixing and matching of different exons either within a gene or between two non-allelic genes owing to errors in meiotic recombination. This process, termed exon shuffling, could lead to new proteins with novel combinations of functions. As an example, let's consider the gene of tissue plasminogen activator TAP or TPA. The TPA protein is an extracellular protein involved in limiting blood clotting. It has 4 domains of 3 types, each encoded by an exon. One exon is present in 2 copies because each type of exon is also found in other proteins. The gene for TPA is believed to have arisen by several instances of exon shuffling and duplication. The TPA also protein slows the clotting reaction and therefore limits the damage that can result from heart attacks and some types of stroke, as long as it is administered immediately to victims. How transposable elements contribute to genome evolution? The persistence of transposable elements as a large fraction of some eukaryotic genomes is consistent with the idea that they can play an important role in shaping a genome over evolutionary time. These elements can contribute to the evolution of the genome in several ways. They can promote recombination, disrupt cellular genes, or control elements that carry entire genes or individual exons to new locations. The presence of homologous transposable elements sequences scattered throughout the genome allows recombination to take place between different chromosomes. Such alternation are probably detrimental, causing chromosomal translocations and other changes in the genome that may be lethal to the organisms. But over the course of evolutionary time, an occasional recombination like this may be advantageous to the organism. The movement of transposable elements around the genome can have several direct consequences. For instance, if a transposable element jumps into the middle of coding sequences of a protein coding gene, it prevents the normal functioning of the interrupted gene. If a transposable element inserts within a regulatory sequence, the transposition may lead to increase or decrease it. Protection of one or more proteins. Transposition causes both types of effects on the gene's coding for pigments synthesizing enzymes in McLintock's corn kernels. Again, while such changes may usually be harmful in the long term, some may prove beneficial. During transposition, transposable elements may carry along a gene or group of genes to new position in the genome, or the mechanism probably accounts for the location of the A globin and B globin gene families on different human chromosomes, as well as the dispersion of the genes of certain other gene families by a similar tag-along process. An exon from one gene may be inserted into another gene in a mechanism similar to that of exon shuffling during recombination. For example, an exon may be inserted by transposition into an intron of a protein coding gene. If the inserted exon is retained in the RNA transcript during RNA splicing, the protein that is synthesized will have an additional domain which may confer a new function on the protein. Recent research reveals yet another way that transposable elements can lead to new coding sequences. This work shows that an allo element may hop into introns in a way that creates a weak alternative splice site in the RNA transcript. During processing of the transcript, the regular splice sites are used more often so that the original protein is made. When occasion, however, splicing occurs at the new weak site, with the result that some of the allo element ends up in the mRNA coding for the new portion of the protein. In this way, alternative genetic combinations can be tried out while the function of the original gene protect is retained. Clearly, this process protect or produce either no effect or harmful effects in most individual cases. However, over long periods of the time, the generation of genetic diversity provides more raw material for natural selection to work on during evolution. Recent advances in DNA technology have allowed researchers to sequence and compare the genomes of many different species, increasing our understanding of how genomes evolve. Thanks for listening.